If something alive had no parents, would we recognize it as one of us? Not as a metaphor. Not as a myth of God shaping clay. I mean a literal, physical living thing that did not come from a mother or a father or any ancestral cell. A cell with no lineage. A creature with no family tree behind it. Would we look at it and say, "Welcome to the community of life"? Or would we say, "You're a machine, an artifact, an imposter"? Every living thing you've ever met, every friend, every tree, every bacterium, has a past that goes back, uninterrupted, for billions of years. You are the current edge of a 4 billion year relay race. Your body is a library of chemical decisions your ancestors made without ever knowing you would exist. Your eye color, your height, your tendency to get sunburned, your family's medical history, these are all echoes of that continuous line of reproduction. Our sense of identity is deeply anchored in this inheritance. We talk about family trees, ancestry tests, genetic traits we got from mom, or inherited from dad. We keep records, birth certificates, medical files, lineage charts. Our entire experience of life is framed inside this story. Life comes from life. So what happens to that story if, one day, in a quiet laboratory somewhere, a group of humans takes nothing more than raw chemicals, lipids, amino acids, nucleotides or their synthetic cousins, and patiently assembles them until, without any biological ancestor, they begin to move, to grow, to divide, to evolve? Would that thing be alive, or would it just be an ultra-complex gadget made out of organic parts? When does chemistry cross the threshold into biology? That's the door we're going to step through together. Because right now, across the world, scientists are genuinely trying to do this. To build life from the bottom up. To make new forms of heredity. To write genomes that have never existed before and watch as cells boot them up like software. They are chasing what we might call a second genesis. When I say, second genesis, I don't mean aliens landing in a spaceship. I mean something more radical, and in a way more unsettling, a separate origin of life right here on Earth. A lineage of organisms whose tree of life does not merge with ours if you trace it back far enough. A completely independent branch of living matter, born not from primordial oceans, but from glassware, pipettes, and human intention. So the central question for this investigation is this: are our current experiments with synthetic cells, xeno-DNA, and artificial genomes actually approaching that second genesis, or are the headlines racing far ahead of reality? You've probably seen the claims. Scientists create artificial life, or life engineered from scratch. They're dramatic, they're clickable, and they're almost always oversimplifications. In this episode of the Science Audit, we're going to slow down and do what we do best: separate what's actually happening in the lab from the stories we tell about it. We'll ask, what even counts as life, in a way a chemist can use at the bench? How close are we to making something that can really evolve on its own? What would a genuine second genesis look like? And what would it mean, for our understanding of the universe, and for our place inside it, if we discovered that life is not a miracle that happened once, but a pattern that can be reproduced? Let's start with the most slippery question of all: what is life? You might expect that by now, science would have a single, precise definition. But we don't. What we have are working definitions, tools, designed for specific tasks. One of the most famous ones comes from NASA. Life, they say, is a self-sustaining chemical system capable of Darwinian evolution. That sounds technical, but let's translate it into something you can feel. Imagine a candle flame. As long as you feed it fuel and oxygen, it persists, it maintains its structure, it even spreads from one wick to another. Life, in this NASA view, is like a flame of chemistry that somehow manages not only to keep burning, but to make imperfect copies of itself, so that over time the pattern can change, adapt, and become more complex. Self-sustaining chemical system means the flame can keep burning as long as its environment offers resources. Capable of Darwinian evolution means it doesn't just reproduce identically, it introduces variations, and some of those variations survive better than others. The winners become the new standard, generation after generation. Other thinkers emphasize different aspects. Some are drawn to a concept called autopoiesis, a beautiful but slightly intimidating word that literally means self-making. In that view, a living thing is a bounded system that continuously rebuilds itself from the inside. Your cells are constantly breaking down molecules and rebuilding them, patching membranes, repairing DNA, replacing worn-out proteins. By this definition, life is like a factory that is also rebuilding the factory while the assembly line is running. Still others argue that life is first and foremost about information. In that frame, metabolism, the chemistry of energy and materials, is important, but secondary. The heart of life is that it stores recipes for making itself, copies those recipes, and edits them through evolution. In this sense, DNA is like software written in a four-letter alphabet, and organisms are the hardware that runs that code. So which is it? A flame of chemistry, a self-building factory, or a running program in matter? Here's the crucial thing. These definitions are not eternal truths carved in the fabric of reality. They are lenses. Tools. Each one highlights certain features and downplays others. And that matters because the lens you pick doesn't just shape how you describe life, it shapes how you try to build it. If you think life is fundamentally a metabolism, you'll try to build a network of chemical reactions that keeps itself going. If you think life is fundamentally information, you'll put your energy into making new kinds of genetic molecules that can be copied and mutated. If you think life is autopoietic, you'll focus on building compartments, membranes, droplets, that can maintain themselves and house the chemistry inside. To understand how ambitious our synthetic efforts are, we need a quick, honest look at how natural life seems to have solved this problem the first time around. This is the story of abiogenesis, how life could have arisen from non-living chemistry on the early Earth. We don't have all the details, and we should be suspicious of anyone who claims we do. But we have plausible outlines. Picture the young Earth, billions of years ago. No trees, no animals, no oxygen-rich air. Just oceans and rocks and an atmosphere laced with gases like CO2, nitrogen, maybe methane and hydrogen. Into that mix you add energy, lightning, ultraviolet light from the sun, heat from volcanoes and hydrothermal vents. Under those conditions, simple molecules can form more complex ones. We know this because we can do some of those reactions in the lab. Amino acids, the building blocks of proteins, can be created from simple precursors. Nucleotide-like molecules, pieces of the letters that make up DNA and RNA, can be synthesized in realistic prebiotic chemistries. You get a chemical soup with many ingredients. Somewhere in that messy ocean of possibilities, a critical transition happened. Certain molecules gained the ability to make crude copies of themselves. Not perfectly. If the copying were perfect, nothing would evolve, but well enough that there were lineages of molecules that lasted longer than others. This is where the idea of an RNA world comes in. It proposes that before DNA and proteins took center stage, RNA molecules might have done double duty, storing information and catalyzing reactions. But molecules alone are not enough. To get something that starts to look like a primitive cell, you need at least three things. First, a container, some way of drawing a boundary between inside and outside, so that useful molecules can accumulate instead of diffusing away. Second, a way to store and copy information, something like nucleic acids. Third, a means of harvesting energy and materials, metabolism, to keep the whole system from running down. A container, a memory, and a metabolism. In human terms, think of it like this: a house, a set of blueprints, and a supply line of food and electricity. You can't really claim to have something that behaves like an organism unless these three are working together. Over time, Earth's chemistry seems to have converged on a particularly elegant solution: the DNA, RNA, protein system that every known organism today uses. DNA became the stable archive, the long-term library. RNA became a flexible messenger and sometimes a catalyst. Proteins became the workhorses and machines, folding into intricate shapes that could perform almost any chemical trick. Out of the absurdly large number of possible ways you could arrange matter to do self-sustaining, evolving chemistry, life on Earth chose one. Not just in general outline, but in specifics. 4 main nucleotides in DNA: A, T, C, and G, 20 standard amino acids. A near-universal genetic code that maps 3-letter nucleotide words into those amino acids. If you zoom all the way out, the biosphere is a gigantic network of branches and leaves, but every branch traces back to this same trunk, the last universal common ancestor, LUCA. And this is where synthetic biology enters the story. Because we've learned enough about these mechanisms that we can now start to tinker. Not just observing life, but editing it, rewriting it, and maybe building it. In 2010, Craig Venter's team announced something that made headlines worldwide. They had created the first synthetic cell. What they actually did is both subtler and, in some ways, more astonishing than the headlines suggested. They chemically synthesized the bacterial genome. Essentially, they wrote the DNA sequence on a computer, assembled that sequence piece by piece in yeast, and then transplanted that synthetic genome into a closely related bacterium whose own DNA had been removed. The host cell, stripped of its original genetic identity, booted up the new genome. It started reading the synthetic DNA, making proteins accordingly, dividing, and forming colonies. From that point on, the lineage of those cells was controlled by a genome that had been designed and built by humans. Later, Venter's group refined this work to create what they called a minimal cell, JCVI Syn 3.0, a stripped-down organism with a genome of just 473 genes, far fewer than most free-living bacteria. The idea was to find the smallest set of genes that could still sustain a living, dividing cell under ideal conditions. This is life as reverse-engineered machine. You remove components, and see how much you can take away before the engine stalls. Impressive? Absolutely. A second Genesis? Not quite. In both cases, the cells themselves, the membranes, the ribosomes, the whole molecular chassis, came from existing organisms. We wrote a new operating system, but we still installed it on an old computer. The cytoplasm, the metabolic networks, the entire evolutionary history encoded in their molecular machinery, that was not built from scratch. Other groups have been expanding the basic alphabet of life rather than minimizing it. Normally, DNA uses 4 letters. But clever chemists have managed to create new base pairs, extra letters, that can fit into the DNA double helix without breaking its geometry. Some bacteria can be engineered to accept these unnatural base pairs, copy them during replication, and even use them to encode new, non-standard amino acids. Think about that. For 4 billion years, all known life has been spelling its recipes with the same 4-letter alphabet. Now, in controlled lab conditions, we have organisms writing with 6 letters. It's as if we taught life a new vowel and watched it start using it in its words. This isn't just a parlor trick. Expanding the genetic alphabet could, in theory, allow organisms to build proteins with new chemistries, materials that life has never explored. And it raises a deep question. If you can change the alphabet and the grammar of life and still have something that functions, then how essential was our particular alphabet in the first place? Meanwhile, other researchers are trying to build cells from the bottom up, what are often called protocells. These are simple droplets made of fatty acids or other amphiphilic molecules that spontaneously form membranes in water. Inside those membranes, you can trap RNA strands or other reactive molecules. Feed the system extra building blocks, and the droplets can grow. Agitate the solution, and they can divide into smaller droplets, sometimes carrying their internal contents with them. It's a primitive shadow of cellular life, but it's a powerful model. You have a container. You have information-carrying molecules. You can even get basic cycles going where internal chemistry changes the membrane, and the membrane, in turn, affects which molecules can get in or out. This is a minimal sketch of that house blueprint supply line trio we talked about earlier. Then there are the xenonucleic acids, XNAs. These are molecules that play the same basic role as DNA and RNA, storing genetic information and sometimes acting as catalysts, but they use different backbones or different bases. One famous example is Hakamoji DNA, Hachi, for 8, and Moji, for letters, an 8-letter genetic system that includes the usual A, T, C, G plus 4 synthetic letters. What makes XNAs remarkable is not just that they exist, but that some of them can be copied with reasonable fidelity by engineered enzymes, and even evolve in the lab. In test tube experiments, XNAs have been shown to undergo Darwinian selection. You start with a pool of random sequences, expose them to some challenge, and select the ones that perform best. Cycle this, and the population shifts. That's evolution, running on a genetic medium that Earth never used. So where does all of this leave us? How close are we, really, to making life from scratch or launching a second Genesis? First, honesty. We have not yet created a fully independent, evolving organism from raw inorganic materials alone. No one has taken carbon dioxide, nitrogen, mineral salts, and sunlight and produced a brand new, self-sustaining lineage that was never part of the existing biosphere. We are, however, pressing against the boundary on several fronts. We can design entire genomes on a computer and watch them boot up inside existing cells. That's synthetically written life, but it still rides on an inherited molecular machine. We can make protocells that grow and divide, modeling aspects of early life. We can produce xenogenetic systems that store, copy, and evolve information under controlled conditions. To make sense of this landscape, I find it useful to distinguish between what we might call synthetic life 1.0 and synthetic life 2.0. Synthetic life 1.0 is what we've mostly done so far, modified Earth life. We tweak the code, rearrange the genome, add new letters, remove non-essential genes. We're building new software and occasionally some new hardware modules, but we're still using the same overall architecture that natural evolution gave us: lipid membranes, ribosomes, ADP, and the DNA, RNA, protein triad. Synthetic life 2.0 would be something more radical, an alternative biochemistry with independent evolutionary potential. A lineage whose deepest ancestry is not Luca. This might run on XNAs instead of DNA, or use a completely different genetic code, or build its membranes from unfamiliar molecules. The key is not just chemical novelty though, it's whether this system can sustain itself, reproduce with variation, and adapt in an open-ended way without leaning on existing life. And that raises some unsettling paradoxes. Imagine a thought experiment. We take lipids, amino acids, nucleotides, salts, all purchased from chemical suppliers or synthesized from inorganic precursors. We build ribosomes piece by piece. We assemble polymerases, membrane transporters, metabolic enzymes, all of it, not taken from cells but constructed in the lab. Then, after a long and messy process, we finally produce a cell that can grow and divide on its own, in a simple nutrient medium. It uses standard DNA, with A, T, C, and G. It uses the usual 20 amino acids and the canonical genetic code. Its chemistry is Earth-like in every way, but none of its molecules have ever been part of another cell. Every single one is chemically synthesized. When it starts to divide, have we created a new origin of life? Or have we made an extremely elaborate clone of Earth life's overall design? Is the second genesis defined by historical independence, no borrowing of molecules, no borrowing of structures, or by architectural independence, new chemistry, new rules? This is like the ship of Theseus applied to biology. If you replace every plank of a ship with a new one, it's still the same ship in some sense. But if you build a perfect copy of the ship next to it, out of new materials, is that a new ship or just another instance of the same design? In our context, is the genesis, the birth of a new design, or the first moment that design is instantiated in the world? Then there's the paradox of continuity. At what point does a reacting mixture become a living system? Suppose you have a protocell-like droplet on a slide. At first, it's just a container with some chemicals. You add more components, some RNA, some metabolic enzymes, some energy source. For a while, it's just chemistry. At some moment, perhaps, it crosses a threshold where the reactions inside start to feed back, maintaining the membrane, changing its permeability, enabling division, and preserving informational molecules. But there is no single spark, no obvious flash of being. It's a gradient. One frame of the movie is non-life. Many frames later, you have something that can evolve. Where in that movie does life begin? During assembly, when all the parts are finally in place? Or only once the system has gone through a few cycles of growth, division, and selection, once it has a past? Now let's push our reality model further. If life is fundamentally information, as many argue, does it matter what physical substrate it runs on? We're used to thinking of DNA as the medium of heredity, but information is more abstract than that. A poem doesn't care if it's written on paper, displayed on a phone, or remembered in your head. The same pattern can move between media. In computing, the same program can run on a silicon chip, a quantum device, or, if you're ambitious enough, through clever arrangements of mechanical parts. The substrate changes. The logic can stay the same. So imagine a biosphere where genes are encoded not in DNA, but in an XNA we invent. Or in magnetic patterns in a mineral. Or in arrays of synthetic polymers that have never existed in nature. If such a system could support heredity, variation, and open-ended evolution, would it be any less alive, because its medium is unfamiliar? Or does life transcend the specifics of chemistry, in the way language transcends paper and ink? And if that's true, then some of our debates about creating life become debates about whether we've instantiated a particular pattern of information processing, rather than whether we've triggered a metaphysical spark. This brings us to the second genesis question in a more precise way. A true second genesis would mean that somewhere, somehow, whether in a warm little natural pond or in a chilly steel bioreactor, a lineage arises whose deepest ancestry does not merge with our tree of life. If you could label every ancestor in its past, you'd never find Luca. You'd find something else, maybe a different starting molecule, maybe a different coding scheme, maybe a different metabolic core. In that sense, xeno-DNA and xNAs are not just clever toys. They're potential pathways toward new trees of life. If we can get an XNA-based system to manage three key things, heredity, variation, and open-ended evolution, then we have something that is not just an extension of Earth's solution, but a different answer to the question, how can matter store and refine information over time? But there are enormous practical barriers between interesting chemistry and genuine evolution. Let me name a few, because they highlight just how special Earth life's solution is. First, error rates. To evolve, you need mutation, but not too much. If copying is too perfect, nothing changes. If copying is too sloppy, the system can't maintain any useful information and falls apart in what's called an error catastrophe. DNA replication in modern cells is astonishingly accurate, with error-correcting enzymes proofreading as they go. Getting a brand new molecule like an XNA to that sweet spot of just enough error, but not too much, is very hard. Second, replication machinery. DNA doesn't copy itself. It's copied by proteins, polymerases, that are encoded in the DNA. This is a classic chicken and egg loop. Early life had to bootstrap from much simpler chemistries, maybe with RNA copying itself or with minerals catalyzing reactions. For a new genetic system, we need either to design entirely new enzymes that can copy it, or to discover simple chemical cycles that can do the job without complex catalysts. Third, energy capture. Life is not just a pattern of information, it is a pattern that resists the universe's relentless drift toward disorder, entropy, by constantly channeling energy. Whether it's bacteria oxidizing hydrogen sulfide at a hydrothermal vent, or plants turning sunlight into sugars, every lineage has to solve the same basic problem: where do I get energy? And how do I use it to maintain my internal order? A synthetic organism that can't plug into some energy gradient in its environment is a fancy firework. It flares up in a lab flask and then would vanish without constant human support. Fourth, ecological niche. For a second genesis to become more than a curiosity, it needs a place to live, a way to coexist, or compete, with existing life. Earth's biosphere is already full of extraordinarily efficient organisms that will eat almost anything that looks like food. A brand new form of life will have to find a niche where it's not instantly digested. These barriers are why, right now, most synthetic systems are fragile and heavily dependent on human caretakers. We can nudge molecules around, but getting them to stand up and walk into evolution's arena on their own is another level. And yet there's a strange, slightly eerie possibility. We might create a simple but robust, alien, lineage in the lab before we ever detect alien life in space. If that happens, if, say, an XNA-based protocell system is coaxed into a state where it can undergo hundreds, thousands, millions of generations of evolution in a controlled environment, we will be witnessing a second genesis in a deep sense. The universe will have learned a new way to remember itself. What does that mean for how we understand reality? If we can create life, or at least a lifelike evolutionary system, in the lab, then life moves, conceptually, from the category of miracle to the category of engineering problem. Not to diminish its value, but to reclassify it as something that emerges when certain conditions are met. That's a philosophical shock. For centuries, life was seen as fundamentally different from non-life, animated by a special vital force. Modern biology has largely discarded that idea, but emotionally, many of us still carry a residue of it. The notion that you could, with enough knowledge and care, assemble a living thing from dust and gas and sunlight, no divine spark required, can feel disorienting. But there is another way to see it. If life is reproducible on a lab bench, if the recipe is not unique to Earth's history, then maybe the cosmos is biased toward life. Maybe the emergence of self-sustaining, evolving chemistry is not a freak accident, but a kind of phase transition that happens whenever the ingredients and the energy flows are right. In physics, when you cool water below 0 degrees Celsius, it freezes. When you heat certain magnets above a critical temperature, they lose their magnetization. These are phase transitions, abrupt shifts in the collective behavior of matter. What if life is a phase transition of chemistry? Wherever the universe provides the right conditions, matter tips into a state where it can store information, replicate it with variation, and let selection do its sculpting work. If that's true, then the night sky might be full of variations on this theme. Different planets, different chemistries, different solutions, but the same underlying logic, matter trying out ways to remember and refine itself. And that brings us back to you, to the most intimate human scale. You are not an exception to physics. You are what happens when physics discovers a way to preserve and refine information across time. Your DNA is a compressed archive of your ancestors' successes and failures. Your brain is a dynamic pattern of electrical and chemical activity that encodes your experiences, desires, memories. In that sense, you are already synthetic. Not in the sense of being artificial, but in the sense of being synthesized, from stardust, from water, from carbon dioxide through countless prior generations of life's chemistry. You are the product of a natural laboratory that has been running experiments for billions of years. If we had a second laboratory, our own, where we intentionally design new lineages of matter that can learn, adapt, and persist, then we become, in a very literal way, co-authors in the story of life in the universe. That power is intoxicating. It is also dangerous. Synthetic life, whether 1.0 or 2.0, carries enormous potential for medicine, climate solutions, and manufacturing. Imagine bacteria engineered to sequester carbon more efficiently, or to produce biodegradable plastics, or to synthesize drugs on demand inside your own microbiome. Imagine resilient crops that can thrive in changing climates, designed with an understanding of minimal genomes and new genetic alphabets. But with power comes risk. Anytime we create an organism with new traits, there is a non-zero chance of unintended consequences. A bioengineered microbe might behave differently in the wild than in the lab. An xNA-based system thought to be biocontained, because standard organisms can't read its genetic code, might still interact with ecosystems in unforeseen ways. This is why biosecurity and biosafety are not side notes. They are central to the conversation. We need rigorous frameworks, technical, ethical, and legal, to decide what experiments are acceptable, who gets to run them, and how we monitor and respond to emerging risks. We've already seen how quickly information technologies can outrun our social and regulatory capacity. We cannot afford to repeat that pattern with technologies that manipulate the fundamental architecture of living systems. Then there's the question of moral status. If we design a lineage of evolving organisms, especially if they develop complex behaviors, or if they cross some threshold of sentience somewhere down the line, what obligations do we have to them? Are they tools, like self-driving cars or industrial robots? Or are they participants in the broader community of life, deserving at least some level of moral consideration? Today, our synthetic cells and protocells are chemically clever, but not conscious. They don't suffer. But evolution is an open-ended process. If we seed a new tree of life and let it grow, we are initiating a story whose later chapters may involve beings with experiences, preferences, maybe even cultures that we did not foresee. When people say, "playing God," they often mean, "You are doing something hubristic, beyond your moral pay grade." It's an understandable reaction. But I think we need a more nuanced narrative than either "we are gods now," or "we must never touch this." One alternative story is co-creation. For 4 billion years, nature has been the sole author of new lineages. Now, with synthetic biology, we are beginning to collaborate, still bound by the laws of chemistry and physics, still deeply ignorant in many ways, but contributing intentional designs into the pool of evolving matter. The question is not whether we should do this at all, we already are. The question is how to do it with humility, with foresight, and with a sense that we are accountable not just to our own species, but to the larger fabric of life on this planet. Because if a second genesis happens on Earth, whether in a natural niche we haven't noticed yet, or in a carefully managed lab, our planet will become a multi-origin biosphere. A living laboratory in which two or more independent answers to the question, how can matter remember itself, coexist, compete, and maybe even cooperate? That would be a profound shift in how we think about life elsewhere. Instead of looking for life like us, we'd be living alongside life not like us, yet still here, under the same sky. Earth would point outward to the rest of the cosmos and say, "Look, multiple origins are possible in the same environment. Don't underestimate the universe's creativity." And yet zoom back down from the grand, abstract view, and consider something intensely personal, your own cells, your own DNA. Inside you right now, trillions of cells are running the standard Earth life operating system. They are reading the same 4-letter code, using the same 20 amino acids, the same genetic grammar that was hammered out long before mammals, long before multicellular life, maybe long before oxygen filled the atmosphere. You are one particular answer that the universe gave to the problem of persistence. Synthetic life, if and when we truly achieve it, will be another answer. Not necessarily better, not necessarily worse, but different. Another way of being a pattern that resists oblivion. So I want to leave you with a contemplative image. Picture a beaker in a lab, sitting under fluorescent lights, filled with a clear solution that, to the naked eye, looks like water. Inside that solution, perhaps, is a protocell system tinkered into existence by human hands. Tiny droplets forming and dividing, strands of xenonucleic acid copying imperfectly, a bare-bones metabolism cycling as long as you keep supplying energy. On the other side of the glass window of that same building, in a hospital or a home, a newborn child takes their first breath. Two very different scenes. And yet, at a deep level, they are both moments in the same cosmic experiment. Can matter learn to remember itself, to anticipate, to adapt, to care? The beaker is an attempt to recreate, in miniature and with guidance, the kind of transition that gave rise to every cell in that newborn's body. The child is the current summation of one successful lineage's journey through that space of possibilities. Both are expressions of the same underlying drive, the universe seeking durable patterns in the face of entropy. So as we draw this journey to a close, let's gather the threads. We began with a deceptively simple, human question: if something alive had no parents, would we recognize it as one of us? To approach that, we explored what we mean by life, in operational terms, self-sustaining chemistry, self-building systems, information-bearing programs. We traced a sketch of how life arose from simple chemistry on early Earth, and we saw how evolution converged on DNA, RNA, and proteins as a particularly elegant solution. We looked at how far synthetic biology has come, rewriting genomes, minimizing them, expanding the genetic alphabet, crafting protocells, and inventing xenonucleic acids that can store and evolve information. We drew a line between synthetic life 1.0, modified Earth life, and synthetic life 2.0, truly alternative biochemistries with independent evolutionary potential. We wrestled with paradoxes. Is an assembled cell using standard chemistry but no ancestral parts a new origin, or an elaborate clone of a design? At what point does a reacting mixture cross the threshold into life? If life is fundamentally information, does its material substrate matter, or is DNA just one of many possible media? We defined a second genesis as a lineage whose deepest ancestry does not merge with ours, and we saw how xNAs and other synthetic systems might, one day, host such a lineage, if we can overcome challenges of error rates, replication, energy capture, and ecological fit. We confronted the possibility that we might create a simple alien biosphere in a lab before we find aliens in the sky. Then we faced the implications. If we can move life from miracle to engineering problem, it suggests that the universe might be fertile with origins, life not as a one-time fluke, but as a phased transition of matter. It reframes our identity. We are not outside of physics, but an expression of it. Our ability to design new lineages makes us co-creators, carrying both extraordinary opportunity and heavy responsibility. And finally, we stepped back to see ourselves and our synthetic experiments as parts of one ongoing question the universe is asking: how can patterns endure and grow in a world ruled by decay? That's the synthesis. Now, a contemplative takeaway for you, personally. Tonight, or the next time you find yourself alone with your thoughts, try this thought experiment. Imagine that, somewhere in a quiet lab, the first truly independent synthetic organism has just been born. No camera crews. No headlines yet. Just a small group of scientists who have been staring at data for weeks, and then suddenly see the unmistakable signal: growth, division, variation, adaptation. A new lineage has crossed the threshold. Picture one of those scientists stepping outside the building, maybe into the night air, knowing what has just happened. Ask yourself, if you were that person, looking up at the sky, how would your relationship to your own life feel different? Would you feel diminished now that life is something we can fabricate? Or would you feel a strange kinship with this new lineage, as if the universe had just revealed another facet of your extended family? Would your own ancestry, stretching back through primates, through ancient microbes, to that first genesis, feel less unique, or more miraculous, now that you can see it as one of multiple possible paths through a much larger landscape? And bring it even closer, what does it do to your sense of self to realize that you are, in some sense, a temporary caretaker of a very long-running chemical experiment, a pattern that began long before you and will, in some form, outlast you? How might that change the way you treat your body, the ecosystems around you, the technologies we're building? Carry those questions with you, because they're not just about synthetic cells in a distant lab. They're about how you locate your own story in the universe's story. And now, as always on the Science Audit, we end not with an answer, but with the next great question, the door that opens onto the next frontier. If we do create a second genesis, if life turns out to be something we can instantiate, like a program we can run in different media, then we will have taken a step toward dissolving the boundary between what is natural and what is made. So here's the question I want to leave you with. If life itself can be engineered, if the distinction between born and built begins to blur, what does that say about the experiences that feel most precious to us, our memories, our loves, our sense of continuity over time? If time, as physics increasingly suggests, is not a simple flowing river but a more complex, perhaps even illusory structure, what does that mean for the memories we hold most dear, those fragile patterns in our brains that tell us who we are? In other words, if we can learn to make new lives, can we also learn to understand, at a deeper level, the life of a moment? What, exactly, are we preserving, when we fight so hard against forgetting? That's where we'll go next time. Until then, stay curious, stay kind to yourself and to the fragile, astonishing chemistry that makes you possible, and I'll see you in the next investigation.